Okay, hi everyone. So this is the microscope part of that lab, right? Remember, it's metric system and microscopy, or metric system and microscope. So now we're going to go over the microscope part of it. All right, so again, you guys have the PDF. Please read through the introductory information that's in the lab. Remember, you're going to be following along with your PDF and writing in or typing in those answers, as I said, in that metric system, or that first part of the lab. Okay. Now it gives you a little bit of introduction about light microscopes. Okay. And so they're called light microscopes because light rays pass through. And if you look here at my microscope, right, there's a light source. Okay. Now, mentions the difference between a dissecting microscope, okay, and a compound light microscope. Please make sure you read the differences. I'm going to expect that you know the information that's presented in terms of the difference between a dissecting and a compound light microscope. Also, what some of these terms mean, right? They're in bold. What compound means: two sets of lenses, okay? Meaning ocular lenses here, right, where we're looking through, and then these objective lenses, okay? So that's where we get the name compound light microscope. And that is the microscope that we'll be using in this lab. This is an example of a compound light microscope, and that compound light microscope is what you'll use in lab. You'll use it a lot, you know, as you move through other biology courses as well, okay? So we're not gonna have to know the parts of the dissecting microscope. We're really going to be just focusing on this microscope here, that compound light microscope. But we should know what the dissecting microscope is. All right. So if you look on page 15 there, that's where I am. You can see there's some micrographs, some pictures there that shows you a light micrograph, right? So this would be it's showing you a blood smear. So that picture you see in panel A is what if I had a, actually I should have did that. I should have got that. If I had a slide of a blood smear and you look through the microscope, you would see something that looks like that. Okay, you can see the red blood cells and the white blood cells. And then what it's actually showing you here are some other micrographs from an electron microscope. Okay, now it talks about electron microscopes on the next page, on page 16. Okay. Again, we're not using an electron microscope. In fact, electron microscopes require a special sectioning technique. They also require a properly trained person to run them, okay? So anyway, we should know what the electron microscope is and how it's different than the compound light microscope. And again, you only need to know just what it tells you here. Don't go looking for extra information or anything like that, but the basic information that's presented in that PDF, okay? Talks about a scanning electron microscope versus a transmission electron microscope. Just know the basic difference, okay? Another term that's introduced in here is something called resolution, okay? So we should know what that is. Minimum distance between two objects at which they can still be seen or resolved as two separate things, right? So if resolution isn't good, if you have two objects that are really close together, they may look like one. If it's really good resolution, two objects that are really close together, you'll still be able to see them as two, okay? All right. And actually that table shows you some differences between the compound light microscope and the electron microscope. You don't need to know those details, just have a basic idea of the differences. Okay, so you should answer those questions there on the bottom of page 16 there, in terms of, it says which two types of microscopes view the surface of an object, okay? Meaning we're talking about between the compound light microscope, the dissecting microscope, the scanning electron microscope, and the transmission electron microscope, okay? All right. Now, if we now move on to page 17, this is now focusing on that stereo microscope, or the dissecting scope. You can take a picture or take a look at that picture that's there. You are not responsible for this, essentially. You're not responsible for identifying out the parts of the dissecting scope, okay? So you can kind of put an X through this, I guess, or just sort of skip over it, but you don't need to label that diagram, all right? But you can kind of take a look at that the reason why it's called a dissecting scope is you could put a specimen underneath there, right? And you can actually be working on it and doing surgery while looking through. The magnification is a lot lower, but you can do that. As you can see with my compound light microscope, you know, you have to put a slide on there. It has to be really thin. There's no room to work underneath there. Okay, so like I said, you don't need to know the parts of the dissecting scope. Okay, and so therefore you're pretty much sort of skipping page 17 and page 18. Okay, that brings us to page 19, and that is using the compound light microscope. Like I said, that's the microscope we will use most often in our courses. So we will need to identify the parts of this microscope. Okay, so just to kind of review that, eyepieces are here, or ocular lens, right? And there is a diagram on the next page here that you are required to complete. You're gonna fill in that diagram, okay? That's part of your assignment. It also says on number 1, and identifying the parts, right? We're going through there, so we should be able to go through these parts and label them on here, but I'm also pointing them out live on the microscope. So the eyepieces, right? It says some light microscopes are monocular, okay, only one, and some are binocular. So ours is binocular. I don't know if you can if you can see that, but, ooh, sorry, let me just turn that. There's 2 eyepieces in ours, okay? So ours is binocular. And it says, what is the magnifying power of the ocular lenses on our microscope? So the answer is 10x, okay? The ocular lenses are 10x. All right, so if we move through here, viewing head basically holds these ocular lenses, that's here. Arm is here, right? This is what's supporting the rest of the microscope there. Nose piece, revolving device that holds the objective. So this is the nose piece. If you see here, it can rotate, right? And that's holding those objective lenses. And then if you look at those objective lenses, okay, you see that they all have different magnifications, right? So for this one that I have on there now, you can kind of see hopefully, is the 4x objective. It's the shortest one, and in here it's marked red. They're not always color coded the same. And then if I turn this nose piece, now the next one is 10x, okay? So the scanning objective, it asks you what the magnifying power is, and the answer is 4x. For the low power, okay, it asks you what the magnification is, it is 10x. If we now move to the next one, that is the high power objective, and that is 40x. The last one that's here is oil immersion, and that one is 100x. We usually don't use that in this course. You'll use it a lot in microbiology, okay? The reason why it's called oil immersion is that you have to put a little drop of oil there because the objective has to get so close to that slide to be able to give that image and that magnification. All right, so I'm gonna go back to the 4x there. All right, so like I said, you guys are gonna fill in that diagram. You will be responsible for knowing those when we have our practical or our lab exam, okay? All right, so the stage. This is the stage. Sorry, 1 second. This is the stage here, okay? It supports the microscope slide. So you can see I have a slide right here that I placed in the stage. And so if you noticed, oh yeah, you can see this is the stage clip essentially. There's different versions of this, but basically holds that slide in place. Mechanical stage control knobs. Those are, you can kind of see them, I'll turn it. Those are here, right? So you can see it moves the stage or the slide side to side and front and back, okay? So you should be able to label those on your diagram. Course adjustment knob is basically when we're focusing, okay? And so that is right here. So course is the big one. You can see it moves this up and down a lot, whereas this is the fine adjustment. It takes a lot of turns to move it, just a short distance. So when you're under low power, it's okay to use the course adjustment, right? Like under the scanning, sorry. When you're under low power, the 10x or the 40x, you want to use the fine control, okay? All right. Light source, already said that. And then base is this here, okay? So that's pretty much it in terms of the parts of the microscope. Now, the next part of it on the bottom of page 20 there describes how you focus in the microscope, okay? And then you're gonna go through a couple of exercises with about inversion using the letter E, and then also we're going to skip over the field of view, but then we're also going to look at depth of field using a slide that has 3 different strings on it, colored threads. All right, so I'm not going to read through all these separate parts in terms of how to focus. I'm just going to give you a little bit of an overview of how to do that, okay? And then like I said, I'll show a video to show you kind of what I see in that microscope with this letter E. So I'll use the letter E slide to show you the proper way of setting this up and getting it in focus. Okay, and then like I said, it then talks about going under high power. The other thing is on page 21, right? That's where you see the inversion part of that. I will go through that and in a second, okay? At the bottom of page 21, it mentions the word parfocal. We should know what that means. Basically, I'll kind of illustrate that and show you that, but basically, if I have the object in focus under scanning, right, the 4x, and it's in focus and I switch my objective to the 10x, it will remain in focus. That's what it means, parfocal. So just so you know. All right, so let me just kind of just show you exactly the way that we should get something in focus in the microscope, okay? You always should start with scanning, okay? You pull back that clip, you slide your slide in, and you can just by eye look to get it kind of in the middle there, right? Where you see it's right under your objective. And then, like I said, you have it under scanning, you're gonna look in, and you're gonna use the mechanical stage control to look in here and get that, whatever you wanna find, right in the middle, right? And then you can use the course adjustment to get it into focus. So let me just kind of show you it out of focus for a second, and then I'll show you it in focus, okay? So again, I have it under scanning. And the other thing is just keep in mind, because this will be part of the inversion part of it, just so I don't have to tell you again or show you again. I put the slide in, and it looks like a lowercase e, like you would see a lowercase e, okay? When you look in the microscope, it's not gonna look that way. All right, so let me show you. I'm gonna turn on my video. I'm gonna share the screen with my phone video, okay? Now I did this before and it works, so let's see. It should work. Tap screen mirroring. Okay. Okay. Okay, so it's mirroring right now. Okay, so we should be able to see that. So what I'm gonna do is I'm going to now show you what I see in the microscope. Okay, so just so you know, this is really the only way of doing this. So, I gotta stand up for that. Okay, so if you guys, I'm gonna kind of flash it for 1 second. Okay, so you guys can see there what I see. You see that letter E that's upside down? It's a little blurry, all right? So now what I'm gonna do is I'm gonna focus that, no, it doesn't even matter. I'm gonna now focus it a little bit better using this coarse focus here, okay? I hope this is showing you guys. Let me see something. I just wanna make sure. Should be fine. All right. So, now I focused it using the course adjustment, and so let's now see what it looks like. Okay, so it's a little more clear, but again, notice, again, I put it in, when I look at it with my naked eye, it's the letter E, the way you would normally see the letter E. What do you notice about what it looks like now? So if you guys go to page 21, under the inversion part, okay? It says draw the letter E as it appears on the slide. So meaning by the unaided eye, it's just gonna look like a normal letter E, okay? Let me do something here. I'll show you what it looks like. 1 second. Okay, and you saw what it looked like under the microscope. So now let me do this. So what it looks like when I just look at it normally is it looks like that. Okay, that's not what it, and so that's what it would normally look like. So that's what you're going to put in number 1, and then it says draw how it looked when I showed you through the microscope. Okay, so that's for number 2. You can answer number 3 on your own. All right, now I'm going to go back to the video and I'm going to do number 4 and number 5. It says move the slide to the right, move the slide to the left, and tell me what you guys notice. Okay, so I'm going to be manually moving the stage. Okay, so let me stop this share. Hopefully I got a lot of resources going on here at once. Get out of here. Okay, so what I'm gonna now do, just so you guys know, I'm gonna take this, the mechanical stage, and I'm gonna move the stage. Like if I'm looking at it with my naked eye, I'm gonna move that stage to the right. But I'm gonna show you, I'm gonna have my video on so you guys can see what happens when you actually are looking through the microscope at that point, okay? All right, so let me do that. I gotta share my screen again with my phone, and I'm gonna show you guys that. Sorry, it takes a second. I don't know why it has to take so long, but it's going, I hope. Oh, wait. Sorry, there it is. Okay, so we're back. So let me now go back to my camera. Back to my video. All right, so remember what I'm gonna do, right? Now I'm manually moving it to the right, and you're gonna tell me which way it moves in the microscope. So let's see if I have a steady enough hand to do this. Most likely not. All right, so I'm moving it to the right. Wait, let me make sure that's true. All right, ready? Here we go. I'm moving it to the right. Moving it to the right. Which way is the E moving? Right? All right, now I'm going to go back. And I'm going to put it back in the middle. And now I'm going to move it to the left. And you're going to tell me, you're going to write down which way it's moving. So now I'm going to move it to the left. So you can see my E there. And now I'm moving it to the left. And which way is it moving? In there. So let's do one more time. It's back in the middle. I move it to the left. Okay. So hopefully we can see that it goes opposite, right? All right. So anyway, you can answer number 4 and 5 on page 21 under inversion as well. Okay. So obviously when you're looking under the microscope, everything is inverted and it's opposite. Okay. All right, the other thing is, I wanted to show you, I mentioned the parfocal thing, but I just want to show you what happens when I switch to higher power. Okay, so I'm going to keep my screen share on. So now what I did is I'm moving it from the scanning, from the 4x, and I'm now moving it to the 10x, to the low power. And I just want you to see what that E looks like now. If I can get it in focus. Where is it? Oh, there it is. Okay, so I'm going to show you what that E looks like. I mean, I'm sure you can imagine what it looks like. All right, so here we go. So now I put it under this low power, right? The 10x instead of the 4x. Let me get it. Okay, so that's what that looks like there. Now, I'm gonna switch it now to the high power. So again, whenever you're using the microscope, no matter what you're doing, you always start under scanning. Get what you want to see in the center, get it in focus, then you switch to low power. Make sure it's in focus, then you switch to high power. You never put that microscope in and go straight to high power, okay? You always need to switch up gradually. All right, so now I'm gonna go to high power. I'm gonna get it in focus, and I'm not moving it, so I'm just gonna show you what we see without me even moving it, all right? And so let's see what we see now. If I can get it. Okay, so if you can, it looks like you see nothing, but at the top, you can see there's like a black thick line. Well, that's all we see of the letter E now, okay? Because it's been magnified so much that in that field of view, because we're on such a higher magnification, we can only just see one little part of that E. We can't see that entire E anymore, okay? So as you move up in magnification, right, going from say the 10x to the 40x, the field of view gets smaller, right? You're only able to see one little tiny part of the E, whereas at 10x, we could see the whole E, okay? So that's one other thing worth mentioning. All right, let me stop the share and go back. Okay, so, all right, so we're back to our lab manual or to our lab handout. Let's flip now to page 22, okay? And so 22 at the top talks about total magnification. And so what is total magnification? It is the magnification of the ocular lenses times the magnification of the objective lens. Okay, so right now we have the high power or the 40x objective there, right? So what's the total magnification if I'm using that 40x objective? It is 40 times 10, hopefully we can do that math, which is 400. So you guys should be completing this table, okay? That's part of it, we're completing that table. So we did that just for the high power, okay? So I started with that just because it was already in here. So under high power, the ocular lens is 10. The ocular lens is always 10, right? Because this doesn't change. What changes is the objectives as you switch them, right? Okay, so for that high power, ocular lens is 10. The objective lens is 40x. Total magnification is 40 times 10, which is 400. Okay, you should be able to complete the rest of that table on your own. All right, so again, now I just flipped it to the 10x, right? The low power is 10x. What is the total magnification if you're using the 10x? What about if you're using scanning? Okay, if you forget what the magnifications are, you go back a couple pages to that beginning, page 19, where it told you to write down the magnification of the objectives, okay? All right, guys. So the bottom of page 22, the field of view exercise, we're gonna skip, okay? But I did just tell you about field of view. We should understand that. Field of view means what you see when you look under the microscope. The field of view is much larger when you're under scanning, under lower power. When you get, so think about what that letter E looked like, just even from scanning versus the 10x, and then what it looked at like at 40x. The field of view gets much smaller as the magnification increases. Okay, so I just want you to understand that concept. You don't need to worry about writing in any of what's on the bottom of page 22. So that where it says observation field of view, you don't need to complete that. You can put an X there if you need to remind yourself of that. Okay, for page 23 at the top where it says conclusions, total magnification and field of view, you should complete that. You can complete that, okay? I don't need to tell you those answers. You guys can complete them on your own. Now, the last part, well, yeah, it's kind of, oh, we're almost done, by the way. The next part is called depth of field, okay? You don't really have to worry about reading through the procedure or anything like that. We should know what depth of field means, and that just means from when you're focusing or you're looking at something, it's this, from the surface of the object down. The depth of field in a compound light microscope isn't big at all, okay? But there is some depth there. And so what we're gonna do is we're gonna put a slide in that has 3 threads on it, 3 different colored threads, okay? And basically we're gonna determine the order of these threads as we move through, right? So you're gonna determine which one is on top, which is in the middle, and which is on the bottom, okay? And so that's illustrating depth of field. So essentially as you're using the focus, right? The focus knob, that's moving that kind of through the object, okay? That you're looking at. So I'll show you that, and then I'll share, I'll do that screen share one more time, okay? Here, and then we'll basically just determine together what was top, what was middle, and what was bottom, all right? So let me go back to that share again. Sorry, it's coming, I think. All right. Okay, so we're back to that. And so what I did was I put in that colored thread slide. I'm gonna move it, right, to get it into the middle. I'm gonna just get it under low power or scanning first, okay, in focus. All right, so now let's look. I'll show you under here. You guys can see those threads. It's gonna kind of be hard. I'm probably just gonna show you this once because you're not gonna be able to see the subtle difference, but you guys can see that there. Let's see. All right, so you see that. Okay, now I'm gonna move it so that I'm kind of focusing in on the yellow one. Actually, let me go under. I'm gonna switch it to low power. You need to go under. Scanning's not gonna help you determine this. All right, so let me show you now, and you can kind of see this. Right, so if you guys look, so actually it looks pretty good, I think. So if you guys look here, maybe you can kind of tell. You can see that the yellow kind of looks out of focus, right? But the blue is in focus. Okay, so what I'm gonna do, and again, sorry if that's not totally clear, but what I'm gonna do is I'm gonna basically, you know, turn the like I said, the fine focus adjustment and determine what's on top, what's in the middle, and what's on the bottom. And again, that's just basically you're doing it as you sort of move through using the focus knob. So like I said, we'll kind of do this together. This is something that you kind of need to do and get the feel for yourself, but I thought it was worth still kind of going over. So for this, if you guys look at that table, you're just basically completing that table. All right, so we want to know which one's on top, which one's in the middle, and which one's on the bottom. So the top is the blue, the middle is the red, and the bottom is the yellow. Okay? All right, so let me stop my share. I gotta share one more time because we have one more thing to do. And then we're all done. All right, so let me stop the share. Okay, guys. Now, the last part of this lab, right, is to actually make a couple wet mounts, okay? And that's the last two things we're gonna do. The other thing that you're responsible for is completing laboratory review 2, which is on page 27. That needs to be completed. For this one, we're gonna start off with the wet mount, okay, on page 24, and this is for onion cells. So it has you do a wet mount for onion cells and also for cheek cells, okay? So what I guess we want to sort of note is sort of basic differences in terms of how what those cells look like under the microscope. And it's going to be a little bit hard for you guys to tell, right? Because I'm just sharing with you. Hopefully we can get the basic idea. And you can label that diagram that's there. So if you look at page 24 where it says onion epidermal cells, just label that diagram based on that picture, okay? If you looked under the microscope and you really studied it and you were able to, you know, look yourself and adjust the fine focus, you'd be able to find the nuclei as well, okay? And so you're not gonna count the number of onion cells or anything like that, right? So you're gonna leave where it says, you know, where it's numbered 1 through 6, you're basically leaving that blank. You're just labeling that diagram on the right side. So figure 2.11 on page 24, just label 1 and 2. Okay, I'm gonna make a slide and I'll just show you how I make it and then we'll just look at it quickly to see what it looks like. All right, okay guys, so if you want to make a wet mount or make a slide of anything, basically you're just gonna take a blank slide, okay? Okay, so you have a blank slide and what I have is just, let's see, I don't know how good my onion was sitting here a little bit, but I have a piece of stinky onion, okay, that I'm just gonna put on my slide, right? Now, if I just put this under the microscope, well, one, we're gonna put a cover slip on top, but if I just put this under the microscope, I'm not gonna really be able to see anything. So you need to add a dye or a stain. Always you need to add a stain of some sort to be able to visualize cells, okay? So like if I took rat brain tissue and I was sectioning it and I wanted to look at it, I would need to put some stain on there really to be able to see those cells, okay? And there are specific stains, there are just general stains. what we're going to use is something super general. I also don't know how great it is because it was sort of older and I pulled it from the lab. But anyway, this is bromo. I think that's what they asked. They told you can use iodine, right? Oh, methylene blue. Oh, crud. I think I took the wrong thing. Why did I do that? Anyway, usually you can, yeah, I don't know why I just use this. This is not what you should use, but let's see if it works. I think I thought I took methylene blue. So you can use methylene blue. That's just a dye, a stain, right? A general stain. You also can use iodine. I have bromothymol blue, which probably is not going to work because this is really like a pH indicator. Not really sure. I just must have grabbed the wrong tube. I was wondering why it looked kind of funny, but now I know why. So let's just see. It might work. Anyway, you put a drop of whatever stain you're going to use, right? On the, sorry. So what I did was I took a drop from using my transfer pipette that I showed you guys before, and I put a drop on that onion, okay, piece of onion. And then I'm going to put a cover slip on top, okay? So this is kind of like the more fun part of the lab. All right, so now I have a little piece of glass. Sometimes they're plastic. You probably can't see it. And I'm going to place it, sorry, on top of my onion. Now, this onion is a little thick. It should be thinner. Usually I would use a razor blade, but I don't have a razor blade. So anyway, I'm going to put it under there. If it looks really crappy and you can't really see anything, I'll spare you sharing my screen. But I wanted to show you how you make that wet mount. So again, like I said, you would normally want to cut this much thinner using a razor blade so that it's not raised up so high. All right, so let's just see what we got here. Again, I got to go to scanning, right, and get it on. Oh, it's actually not too bad. Let's see what we can get here. I got some droplets kind of on there, a lot of air bubbles, but let's find a spot where we don't have as many. All right, let me see if I can get it if I use low power. Yeah, all right, so I'm gonna show you guys. It actually worked out kind of well. Okay, so let me share again. Actually, you know what I'm gonna do? I'm gonna make the slide of the cheek cells before I share, then that way I can just end by sharing once. So here's my onion, right? I'm gonna leave that under there, and then I'll show you guys that in a second. And now let's make another wet mount of cheek cells, right? So if you guys look, that procedure is on page 25 at the top. And so same thing, see that diagram, that figure 212? You should label that, okay? And then you don't have to worry really about the procedure because I'm doing it, right? And talking to you about it. So basically what you're gonna do, hey, do they want you to take a piece of water? I don't think you really need to. So I'm just gonna take a toothpick and I am going to scrape the inside of my cheek, okay? All right, gotta make sure you get in there and you get stuff on there. And so you may have done this before, right? It's possible that you did this in high school or what have you. So I'm gonna put a drop of our bromothymol blue, which isn't really ideal for a stain, but it should work. Okay, so I put a drop on there just like I did with the onion, right? And now I'm gonna put a cover slip on top. All right, so cover slip on top. I'm gonna kind of push it down so I can try to get rid of some of the air bubbles. All right, and I'm gonna leave it on the side here. We'll see what it looks like. Okay, so now what I'm gonna do is I'm gonna share my screen. So I'm gonna show you what those onion cells look like under the microscope based on the wet mount that I made, and then I'll show you my cheek cells, and then we're finished, okay? So to the best of your ability, fill out this table. And just in terms of kind of what they look like, you can use the pictures really more than what you see from my phone, but I want to show you what it looks like live. Okay, so you should be able to fill in that table. We're not going to look at euglena. I don't have any euglena around. If you're, we're also not going to examine pond water. But again, you're responsible for filling out these review questions. okay? So I told you throughout what you should be filling in through the lab. You also then need to complete the review questions. That's the last part. All right, so let me end by sharing my screen to show you what our onion cells and what the cheek cells look like. And again, you have the pictures to help you with that as well. All right, guys, so last time I'm gonna share, and that'll be it. So let me go, sorry, back to this. Hello. Oy. All right, there we go. Okay. All right, so this is these are the onion cells. Right now it's under scanning, right? So under 4X. So you can kind of take a little look at what this looks like. And so those round circle things that you see are air bubbles. It's going to be kind of hard, like I said, that's why I said use the pictures to describe. But you're not going to really be able to. And I also can't. Let's see if I can try to figure out how to hold this more steady. 1 second. There we go. Sorry, bear with me. You guys are always better at this. My students always take pictures like this. All right, so that's it under scanning. Looks kind of weird. Looks a little dark, and because it's thick, it kind of makes it hard to see this. To see the cells. A thinner section is better because the stain can really get in there. Let me just switch to low power and show you. Again, those really dark round things that you saw, those aren't the cells. Those are just air bubbles in there. But let me see if I can kind of focus in on one area. The cells, as you can see in the picture in your lab, are square, right? Okay, square looking, rectangular, something along those lines, right? And you can kind of see that in there actually. Yeah, so if I get it, let me get a better, come on. Right, so you can kind of see if you look closely. Ah, darn it. What happened here? I lost my, anyway. So you can kind of see little squares in there, kind of, sort of, okay? So those are the onion cells. Like I said, you weren't gonna be able to see that much. Let me just switch it now to the cheek cells, and let's see if we can see those as well. But again, I wanted you guys to see how to make that wet mount. I may not even have anything here. Let's see. I got some. All right, so I have it under scanning right now. I'm just gonna show you what it would look like. It's not gonna look like much. In fact, I probably gotta turn the light down. Let me just turn the light down a little bit. See that? Those little spots? Those are the cells. So let me now go under low power. All right, so now I'm under low power, which is, we were at 4X, now we're at 10X. You see those spots are much bigger, right? Oh, darn, I had it good. See right there where that pointer is? There's a picture of there's a cell there, right? Okay, guys, so that is pretty much it. Hopefully, you know, we got enough. I think we did. You know, obviously not the same as doing it yourself, but, you know, I simulated it for you. So remember, we will have a lab exam, lab quiz that's going to cover this, all of the stuff that we went over, okay? So make sure for your assignment, remember for this lab, You have a metric system assignment, which is based on that handout. There's just some practice problems. Then the other assignment that's metric and microscope lab is what we just went through, remember, in the two videos. So for this video, I just did the microscope. There's another video where I did metric. You're filling in the entire lab and then completing the review questions. All right, thanks guys.